Target acquired. Eliminating contamination.
This video is going to be about what tumors fear, because once it's done, it's not recovered. There is no recovery possible for it once the process is finished, and it is the purpose of it is to disable the CD70 on the tumor and to stop the CD70-27 axis, because if it's ongoing or too long, it leads to ongoing NFKB and then eventually T cell loss that is difficult to bring back. So the CD70 on the tumor has a poly A tail on it, and once it's trimmed away, it pretty much disables the CD70, and that's what the tumor hates or fears. And the tumor will try to stop the trimming of the poly A tail with these caps at the end. Okay, so the two mechanisms, the two main ones here are TTP binding the ARE, and then possibly the main part of the trimming process, CCR4-NOT. Another critical step here, in order to stop the prevention of the TTP binding to the ARE, is to reduce reactive oxygen species, or ROS in short. And that's pretty easy with like a vitamin C, vitamin E, berries. Berries have a lot of antioxidants, possibly other foods and supplements as well. Also, the phosphates, which are inhibitory on the TTP, have to be removed as well. So that goes with supporting PP2A. PP2A activity removes inhibitory phosphates. Another step is to lower P38MK2 signaling, because this pathway prevents TTP phosphorylation, since the TTP cannot be phosphorylated, and PP2A removes the inhibitory phosphates. So the first food here that could help, which is a nut, is pistachios. So it has a tocopherol, lutein, and polyphenols. So it reduces oxidative stress, restoring TTP-ARE binding, which is good, so that improves the CCR4 and NOT recruitment efficiency, which is the irreversible machinery or part of the process that the tumor fears. The next one is really important because it prevents the phosphorylation of TTP, because TTP has to be dephosphorylated, and that is Pecans. And the way Pecans does that is by reducing P38MK2. Now, Pecans do this very strongly, or Pecans are a strong food to prevent the phosphorylation of TTP. Now, Pecans have ellagic acid, also proanthocyanidins. Okay, next one is macadamia nuts, important as well because it increases or supports PP2A enzyme stability. The PP2A is important to remove the inhibitory phosphates from TTP, because if those inhibitory phosphates are not removed, then the poly A tail trimming on the CD70 will not be able to go, or will be disabled, or will not proceed. Therefore, the CD70 oppression on the CD27 on the immune cell will continue. So macadamia nuts help to trim the poly A tail. Macadamia nuts also have oleic acid, palmitoleic acid as well. So macadamia nuts promotes what's extremely important, which is the dephosphorylation of TTP persistence. So if that TTP dephosphorylation is persistent, that will allow the CCR4-NOT to continue the irreversible path that the tumor fears. The next nut is pretty important as well, and that's peanuts. What peanuts do is that it stops or suppresses the MK2 pathway that stops or prevents the dephosphorylation of TTP. So peanuts, they also support the TTP shuttling to cytoplasmic decay, which sounds like a good thing to lead to the poly-A tail trimming. Eventually, peanuts are better coupling to PAN2, PAN3, and then leading to CCR4NOT. PAN2 and PAN3 is beyond the scope of this video. The next nut is rather important as well, and that is almonds, because almonds reduces reactive oxygen species. Lowering reactive oxygen species is critical because ROS blocks TTP-ARE binding. If the TTP cannot bind to the ARE, then the main part that the tumor fears, which is CCR4NOT, will not be able to proceed or to undergo the process that will eventually lead to the poly-A tail trimming on the CD70. Now, almonds will eventually lead to faster poly-A tail collapse, which is a critical and a very good thing in the battle or fight against cancer. Now, the next nut here is cashews and has magnesium and copper. Magnesium is important for lysosomal membrane peroxidation repair, or to put out the fire in the lysosomal membrane. Okay, the magnesium is critical because it could prevent the CD70 on the tumor CD27 axis in the first place, because magnesium supports the lysosome so that the lysosome is functional, so that it degrades the exosomes before the tumor receives the exosomes. Because once the tumor gets the exosomes, that leads to the CD27 CD70 axis. So copper in magnesium supports redox enzymes that control reactive oxygen species. Because if there are reactive oxygen species, then the CCR4NOT binding will not occur. Cashews also improve TTP CR4NOT binding strength. So that's critical to eventually lead to the decapping on the poly-A tail, because the caps at the end are put there to prevent the trimming of the poly-A tail, which is not good. Also, magnesium in cashews also has another role of supporting phosphate removal activity by PP2A, because TTP has to be dephosphorylated, or else the CCR4NOT will not be able to occur. So magnesium has multiple roles in battling cancer. So lysosomal membrane peroxidation prevention, improving dephosphorylation activity. So another blocker of the trimming is the caps, but basically what's needed to remove the caps or decapping is something called DCP1/2, and DCP1 and 2 have to be recruited, and that's what the next one can do, which are sunflower seeds. So sunflower seeds also lowers the P38 MK2 signaling pathway. That's important because MK2 prevents TTP from being dephosphorylated. So sunflower seeds leads to rapid cap removal after tail shortening. Next one here is pumpkin seeds with zinc, and stabilizes RNA binding zinc finger proteins like TTP. So that's good because the TTP has to bind to the ARE. So pumpkin seeds enhances ARE affinity for TTP. And the next one here is pretty good, but these nuts are bad and excessive, and that is Brazil nuts. Has selenium and selenoproteins, and selenium increases glutathione peroxidases, leading to strong reactive oxygen species suppression, which is critical because ROS directly blocks TTP-ARE binding, therefore blocking the CCR4NOT that needs to proceed. So that's it for the video. Like, comment, share, subscribe, and I'll see you in the next video.